This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 97, recorded on October 15th, 2015. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent.
And Daniel Griffin.
Hello, everybody.
How are you guys doing here?
Doing good.
You well?
I'm doing well. Today's been a very busy but interesting and satisfying day so far.
Yeah, same here.
Daniel, this morning a sailboat was going up the river. A little guy, maybe 20 feet long, single sail. That guy's still out there. It's October.
This Saturday I'm planning for a big
yeah?
sail. This is when we get the best winds.
But when it's freezing and snowing, you don't go out there, right?
I sail year-round. On Sundays through the winter, we drop the boats in, and as long as we can get out to open water, we race sailboats every Sunday.
So you lift them out of the water.
We lift them out of the water for the week and keep them stored, dry sailing they call it. And then on Sunday about noon, we drop them in.
Nice.
And we sail them. And actually this year, for the first time, my 15-year-old daughter is going to be skippering her own boat in the racing series with her friends.
That's exciting.
So there'll be maybe somebody I can beat, I guess.
Dickson,
so I trout fish until I can't feed my. I trout fish until I can't feel my feet. When the water gets that cold that you can't feel your feet after 20 minutes of standing in the river, that's the time.
So
the
in the
season's
winter,
over.
don't the trout go to the bottom?
No, they still feed, but they're more lethargic. True, their metabolic rates are lower.
Could you fish all winter?
Yep,
if you
I have
bear
done
the
it.
pain.
I've done it already.
Okay.
Yeah, you could.
Now,
In Utah, I used to fish year-round. I start nymphing.
That's right. Yeah, it works.
Well, my hobby is podcasting, and I do it year-round, no matter what the weather. And today we're doing a podcast called This Week in Parasitism.
Indeed.
For which we first need to start with this amazing coincidence, which last week was the Nobel Prizes in medicine were given out for parasite work.
This is all true.
I was in Germany when this happened. When you heard, Dickson, were you just very happy?
I was crying with joy because I know one of the recipients, and I know him very well. In fact, I've published with him, and I've laughed with him and cried with him and spent lots of time at meetings with him. And I know the full story of the discovery of the drug that you're going to mention, I'm sure. And your wife knows him very well, too, because she used to work over there as well.
Yeah, he was at Merck when she was there.
Yeah, so William Campbell is the man's
He was
name.
one. He shared it with Satoshi Omura for ivermectin.
That's right.
So they split half of the prize.
They did.
And then
1/3 each.
The other half... No,
or one quarter each.
And then the other half went to UU2.
Yeah, that's more controversial.
For artemisinins.
More controversial.
Which are anti-malarials. Why is that controversial?
Well, I heard an NPR show on it about the day after they gave out that prize.
Yeah.
And there was a physician who was involved in, like, before Doctors Without Borders, he was in groups like that. And he spent a lot of time in Vietnam, just after the Vietnamese War. And heard about this new drug that was being worked on in China, but remember the Cultural Revolution was in progress at that point. And I don't know if anybody remembers, but as soon as the United States left Vietnam, which was 1976, China invaded Vietnam. And they were repelled. So when you are in those situations, it was estimated during the Second World War that more people died of malaria than died of bullets, which is incredible to think about, but it's probably true. So here's this drug being worked on, and it was worked on in the basements of buildings throughout China by scientists who were not allowed to call themselves scientists, because if they said, I'm a scientist, you got sent out to the farm. Eventually, this guy said he heard through the grapevine that Mao Zedong heard about the work going on and said, these scientists are exempt from the Cultural Revolution. And this woman was one of those people. But she wasn't the only person. And according to him, she wasn't the dominant researcher. But maybe she's the only one that survived up to this point, so they had to give it to somebody. Because that's a great drug. I mean, it's a wonderful, wonderful drug. whose time has almost gone because animals and humans are now becoming resistant to artemisinin, which is too bad because
the parasites are becoming resistant. Right?
Yeah, I meant to say that.
So we're moving onward.
We are, but I don't know of any good examples of resistance to ivermectin because it's such a long-lasting drug and it attacks such a specific target that I don't think you're going to get that happening there.
So what is ivermectin or the avermectins used? They've made, actually, I think the original compound out of the culture was an avermectin.
That's right.
Then I asked my wife, Doris Cully, who worked at Merck on this, and she said they made derivatives. The chemists at Merck got their hands on it.
That's right.
And I said, why? She said, well, the original compound was very active, but they wanted to get their names on the paper, so they made derivatives.
It's interesting motivational work.
And I said, well, why was Campbell given the prize? She said he was the team leader of this. So there was a collaboration between a Japanese company, Kitasato, of which Satoshi Omura was part.
Yes.
And the organism was obtained from soil in Japan. It's never been isolated anywhere else.
This is exactly right.
And they grew cultures, and they found that it was inhibitory to, what did they use? Helminth initially.
Well, they used Nipostrongylus. And so that screen was set up by Bill Campbell. He actually invented the parasite screen, and that's why he was given credit.
And so they came to Merck and said, let's collaborate because you guys have the screens and the chemistry.
Exactly.
So they gave him the culture.
So then, after that, I think there's a rich history of this drug also beyond discovery. And that is the fact that it became, I think, the first drug ever given away free.
That was for river blindness.
That is exactly right. Yeah. And the person responsible for that was Bill Bradley, who was the senator from New Jersey, who approached Merck as a humanitarian act of kindness on the part of this pharmaceutical company that had already made, according to Bill Campbell, about $10 billion in animal sales.
$10 billion?
Yeah. So they could probably afford to.
So this is used for, it's called HeartGuard and for animals, right? For
it's Mectizan. It's called Mectizan.
Mectizan for animals?
That's right.
But I think it's called HeartGuard.
No, you're right. HeartGuard.
I think HeartGuard.
HeartGuard is
like
one of many.
Isn't it like a brand? Isn't HeartGuard?
Yeah, that's right. It's
like
for Dirofilaria immitis in dogs, which is a filarial parasite.
Dirofilaria, that's right.
So the Mectizan program was the giveaway. It was the giveaway program for West Africa because that's what. But there's only one country that refused to participate in that program. Take a guess.
In Africa?
Yeah.
I'm going with Sierra Leone.
You're going with Sierra Leone. I would go with Sierra Leone.
Sierra Leone?
That's the country that doesn't allow anybody in their borders because they don't trust anyone. They're the ones that still perpetuate the Ebola outbreak.
And so for the Ebola, they did not allow people inside their country?
That's exactly right.
Not initially. They eventually did.
Eventually they did. Now maybe they'll come on board for this because one of the conditions for accepting the drug for free is that you will do the statistics on who doesn't have it and who has it still. And for that to happen, you've got to do epidemiologic surveys. And Sierra Leone doesn't do that. And that's tragic.
Yeah. But I should jump in with the resistance because we did an article recently. If we all remember the one where there's this new, it's not a monoclonal antibody. It's a small molecule inhibitor for river blindness.
Right.
And one of the reasons they were saying that we need to start looking at new drugs is that we're actually, unfortunately, in Ghana, we're starting to see some resistance to ivermectin. We've had these wonderfully successful mass drug campaigns, but when you use that much drug, we're starting to see what the veterinarians saw ahead of us. We're actually starting to see resistance.
In people?
In people.
Okay.
In people. So there was a PLOS article about 3 or 4 years ago, which was the first. And that's what sort of got people a little bit scared about these mass drug campaigns are going to be great if the drug we're using stops working, not so great. Yeah. And the veterinarians were already starting to see this because, as we mentioned, the vets were starting to see it.
But, you have to give this drug once a year for 20 years in people because the worms live for 20 years and the drug doesn't actually kill the adults. It sterilizes them temporarily. So they will recover their ability to make microfilaria within a year if you don't give the drug again. So that's a remarkable program that has huge amounts of follow-up and huge amounts of keeping track, so to speak, of what you're doing. I have deep empathy and sympathy for everybody involved in that program. But look at how many lives the drug has saved, and I think that's why they gave it.
So, in addition to Onchocerca, anything else that's used in people?
Oh yeah, it's used for lots of things like strongyloides, stercoralis. It's used for hookworm. It's used.
We
for...
may even discuss something today for which it can be used.
We might even do that.
No, it's actually it's a drug with a lot of activity. It works against a lot of human and animal pathogens.
Right, and what does it do? I knew someone who worked on the mode of action, but I think your wife did.
It binds a glutamate-gated chloride channel. Which,
there you go.
And that was discovered by Doris Cully, my wife.
There you go.
And the glutamate-gated chloride channel is very unusual.
Yeah.
So that's when she went to Merck. That what's his name? You know him,
Dimitri.
No, this British guy was her boss.
Oh, Mervyn Turner.
Merv Turner.
Yeah.
Was work. They wanted to figure out the mechanism of action, so she started working on that. And she identified the receptor, basically, for it. It's very interesting.
Someone asked me once to describe the chemistry of ivermectin, and I said its common name is dihydro chicken wire.
Yeah, dihydro chicken wire, yes.
We've had Looks very complicated.
We've had that before on TWiP. And then they found that Drosophila are sensitive. They have this channel.
That's right.
And they are sensitive.
That's right.
So they decided to use Drosophila to try and find next generation versions. But
it's good for scabies, by the way. This drug even works for scabies.
Yeah, it's nematodes and a lot of arthropods.
A lot of invertebrates, that's good.
Yeah, invertebrates, not just scabies.
Because if it has a nervous system, it probably works against them.
So have you spoken to Bill since he had this on?
I didn't speak to him, but I emailed him many, many times. And one of my emails, which I delight in just being able to say it, because very often people who get the Nobel Prize are not well known or well liked. In this case, he's well known and well liked. And I said, on the rare occasion, nice guys finish first.
He didn't answer you, though.
Oh, yeah, he did. Yeah, no, he did.
So we should say that we did ask him to come on TWiP over a year ago.
No, he'll be overwhelmed with requests.
Over a year ago.
I know.
Before he got this prize.
No,
he declined us.
He did. Politely and respectfully.
But why would he do that? He has a huge history he could talk about.
You got to ask him. He's not comfortable under these situations.
Now he's going to be even more.
No, he's going to be even more uncomfortable. No, he's totally aware of that one. He knows that's going to happen to him.
I have a book which came from Merck. It's parasitology. It's an old parasitology text.
Yeah.
And he signed the inside. This is Bill Campbell, R80 something, whatever building.
He wrote the foreword to my book, People, Parasites, and Plowshares.
People, Parasites, and Plowshares.
Yeah, he wrote the foreword.
Well, you got him to do that. Now you should.
He loved doing it. He was delighted.
That's going to propel the sales of that book.
Oh yeah, we went from 2 to 4.
Forward by Nobelist William Campbell.
That's true.
So Dickson, when are you going to be around for the Nobel?
I wouldn't go there. Ignoble, yes. Noble, no.
Okay, very good. Let's move on to last week's case. Right. Daniel, remind me what was going on here.
Yes, this was a consultation that was requested by the OBGYN service in the hospital. It was a pregnant woman. She'd been admitted to labor and delivery. She was about to give birth. This was the first pregnancy. And everyone was concerned about this husband's skin problem. It was the skin problem that he had going to be a problem for the wife and the coming baby. And as we noticed, there were no lesions on the woman. The husband had recently developed what he describes as a very itchy skin problem. It's bilateral. It was on his hands. It was involved the skin between the fingers, the web. And he had told us that he travels quite often for business and stays in cheap hotels. He says the beds are often not clean.
Right.
He had his last trip about a month before the symptoms appeared. Yeah. And even though he covers mostly the Northeast, he does have some Midwest travel. And that was where the last trip was. He told us a little bit about some family history. He sells things. We didn't really get specifics on what exactly he sells. There's no animal exposure. And then we had seen, when we examined this gentleman's hands, that he had these small papules on the webs of his fingers. There were brown lines, and there were blood clots at kind of the end of these lines, and he had obviously been scratching quite a bit.
Sounds familiar.
And he had no other skin areas were involved.
Right.
We had a bunch of guesses on this one. Jerry wrote scabies. Nancy writes scabies. I mean scabies. I hope the gentleman contracted them via a freshly contaminated, unlaundered hand towel. Thank you for all you do, guys. Steve writes scabies. Shouldn't have read that. Now I'm itching all over. Can you read, Dickson?
I can't.
You can't. No.
I mean, I can read, but I have nothing in front of me to read.
I can. Well, we could share my computer. I'll read, and then you can read.
This would be delightful.
That sounds.
I'd love that.
Oh, this is going to be great.
The next one, Dickson
True act
should
of
read.
cooperativity.
The next one, because it has
Robin writes, and I passed this.
It has
to
a
Dickson.
poem in it.
Oh my goodness! It has a poem.
Robin.
So here is
Robin.
The old Sarcoptes. That's what he thinks it is. The 7-year itch, ha ha ha, because over a period of years the symptoms subside unless immune compromised, in which case it may be Norwegian, but not a rat. Well, and with apologies to Ogden Nash writes, or no, I guess he must have written this, little mites have upon their backs little mites to bite 'em, and littler mites have even littler mites, and so on ad infinitum. Well, that's not an apology to Ogden Nash, it's to Jonathan Swift, who wrote, little fleas have smaller fleas upon their backs that bite them, and smaller fleas have littler fleas, and so on ad infinitum. So you got the poem right, but the poet wrong.
I'm sure.
Ogden Nash was famous as saying, the Lord in his wisdom invented the fly, and then forgot to tell us why.
Daniel, can you take the next one, please?
Yes. Christine writes, hi, Vincent, Dickson, and Daniel. Greetings from increasingly warm Brisbane, where it is 29°C at 5 p.m., heading towards 33°C tomorrow. Clear skies and a light breeze. Today's case is a scabies infection/infestation caused by the female Sarcoptes scabii mite burrowing into the skin to live and lay eggs. It is infectious through contact, commonly sexual, although it does survive up to 3 days without human contact. Unwashed linen or even cold, washed, and dried may harbor these beasties, so an extramarital sexual encounter is not necessary. Treatment of the patient is recommended due to discomfort and transmissibility. He should be treated with ivermectin or permethrin.
That's right.
Crotamiton or Lindane creams. An antihistamine is probably a good idea for symptomatic relief. Ivermectin is not suitable for pregnant wives, so close monitoring may be the best option for her. Prolonged contact between the man and his new baby is highly likely, so treatment should be started as soon as possible. Yeah. Scabies in the newborn and infant is more widely spread over the skin, including the head and face, and can lead to large blistering lesions. Thank you for your ongoing efforts, Christine from Brisbane. Right. And I saw that there was some cringing from Dickson. I'll address what may have made him cringe later on.
Good.
Peter,
we'll keep reading for now.
Peter writes, diagnosis equals scabies. Wink writes, salivary pharmacopoeia, which was the title of that episode. I have 5 items for you. 1, first, an appreciation. I subscribe to 25 podcasts, and TWiP is my favorite and the one I play first. Thanks.
Thank you.
2, great paper with fascinating, overlapping, interdisciplinary, and pragmatic elements to it. That is the one where the components of the sandfly saliva skew the immune response, right?
Yes, in favor of leishmaniasis. That's right.
3, my guess for the case is scabies, and that the consult was generated out of concern for both the expectant mother and the medical staff.
And the medical staff particularly.
4, I think I heard this morning that Nobel Prizes were given for the discoveries of ivermectin and artemisinin. Hopefully you will discuss this on the next podcast. We did. 5, a suggestion, try doing your searches on DuckDuckGo. Less commercialism and quicker information than Google. Wink in Atlanta. Is it as good, though? I'm not familiar. I mean, I've heard of DuckDuckGo, but I don't use it, so let's try it out with Racaniello. How about this?
Maybe our listeners can try it when they try to figure out today's unknown. They can do a little DuckDuck
trial balloon.
Go Google.
Yeah,
it.
And then the first hit on Racaniello. The Racaniello family was a prominent family of Umbrian origin, which provided a number of administrators in Todi. This is not me.
No, that's
but then
not
the rest of theirs are okay. Did you know there's a Dr. Annette Racaniello in Brookhaven?
I had no idea.
It's not probably related, although I don't know her. Sports medicine. Wow, cool. Anyway, that's DuckDuckGo. Thank you, Wink. Daniel, can you take the next one?
I can. I was just, duck going myself.
was-
But
Trying to duck out of the conversation, so to
and
speak.
where are we up to? Are we
Elise.
We're up to Elise. Okay, Elise writes, "Dear TWiP trifecta, greetings again from Lower Manhattan, where everyone is pleased that Hurricane. Can you pronounce this for me? Joaquin. Okay, thank you. Didn't land and create huge floods. Today it is bright and 66 degrees or 18.8 C. I do have a diagnosis for the soon-to-be father from TWiP 96, but first I must express some fear and consternation at the suggestion in the most recent episode that you will not be producing more episodes of TWiP after you hit 100. Please say it isn't so."
We're just joking.
It's not so.
Yeah,
I love the joke.
He does. He
Well, though
teases
Dickson wouldn't care if we stopped it.
Well, that's exactly right.
I would.
No,
I would care. I would miss this
we
time
tend
spent
to
together.
Josh, we over exaggerate.
Yeah, we're going to continue.
Of course we will.
As long as we're able.
What else would we do?
And if one of us goes prematurely, the other two can continue.
We have no option here. We have to keep going
Unless
forward.
it's me, in which case you don't know how to record. So
You're going to be fine.
You're done. Right. I try to be careful.
All right, my plea made. Here's my diagnosis. I believe that the patient has scabies, an infestation of Sarcoptes scabies, human variety.
Or
I
scabii.
make this diagnosis mostly on the basis of the man's symptoms. This man's infestation conforms to standard symptom description: small papules on the skin, brown lines that are likely the burrows of the mites, a concentration of the papules on the hands and webbing between the fingers, and terrible itching that gets worse at night. Does the itching get worse? Then, because mite activity goes up during the sleeping hours? The mites that cause scabies are extremely contagious. So in hindsight, it is actually very reasonable that the patient's wife, OBGYN, would be concerned about his wife and new baby contracting the rash.
Right.
There was some talk about whether the patient might have contracted this rash through extramarital sexual contact. And while it is true that scabies are likely shared between and among sexual partners or other instances of close physical contact, it is common in nursing homes and among children as well. It is also true that scabies mite infestations can occur from infested towels and bedding. If the patient really has been staying in hotels that don't change the bed sheets between guests, then some opportunistic mites could have found him. There is very good chance that the patient has infected his wife, and they should both undergo treatment and clean the house thoroughly. Vacuum everything. Wash everything that isn't vacuumed in hot water. I didn't see that there is at least one of the topical ointments for scabies that is safe for pregnant women and young babies. So it may not be risky treatment, only a time-consuming one with all that cleaning, which will be a huge drag on top of having a brand new baby at home. As always, thank you so much for everything, and please don't stop broadcasting. Many best wishes.
Can you take the next one, Dickson?
Andrew writes,
"Dear Paratenic hosts," I say Paratenic because in a way you carry us, the audience, with you. That's very nice. Although perhaps Paratenic isn't appropriate because the definition implies that there is no development of the parasite, which is surely not the case for the listeners of TWiP. As I listened to the latest case history, I began to experience a strange paresthesia. I had the distinct sensation my leg was being pulled. In any case, I'm sure I'm not going to cause anyone to faint with amazement when I offer the diagnosis of scabies. If it turns out not to be scabies, again, then I offer an alternative diagnosis of lupus. Fans of Dr. House will understand 'tis never lupus. All the best from a rapidly warming New Zealand in both the short and long time
That's
scales.
wonderful.
That's great.
So down there in New
That is
Zealand
great.
and Australia, they're moving into their summer,
Yeah.
right?
They are, and in fact, this is the perfect time to think about trout fishing in New Zealand. It's a fantastic activity.
Why don't you go?
I've been many times.
Go now. Go tomorrow.
See ya.
Go.
I'm leaving.
So I don't watch House. I never have. What's
Oh, I've
this? It's
watched
never lupus.
Lupus is their first diagnosis because it's such a complicated disease and it manifests as, but it's never lupus ultimately, of course, because that would be too simple.
Daniel, you never watched House, right?
I haven't, but I feel like I should watch an episode.
No, you don't
watch it all the time.
Okay.
No, I mean,
And
it's all
classically
fake
they get it wrong no matter
it's all
what.
fake medicine, right?
It's not fake medicine. No, it's actually they have good advisors.
Do they have physicians as
They do, but some of their cases are so exaggerated that it gets to be commedia dell'arte rather than.
You know, the problem is if you just went into what a normal place sees, it would be boring, right?
Not if you were the victim. No, it's exciting enough in the ER, don't you think?
You know, a lot of it depends where you are.
That's true.
You know, different
New York
places.
on a Saturday night in the ER is the most exciting place you can ever be. Stab wound victims, shooting victims.
Yeah, but that's not interesting. It's tragic.
That is
heart
that is
attack.
tragic.
No,
You know, I have to say,
when I say interesting, I mean riveting.
you know, I have to say, I've been emailing a little bit with some of the infectious disease colleagues with whom I spent time down in Lima, Peru, and just joking about wouldn't it be great if it was always that diverse, the things that you see. But now, you know, you see a lot of the same.
Yeah.
All right, David writes, Dear TWiP Trio, as Dickson said, what a propitious event. As for the case of the husband of the pregnant woman, I will venture a guess that the husband has scabies brought on by the parasitic arthropod mite Sarcoptes scabii. This parasite is a frequent and unwanted guest in cheap motels, and as mentioned on the previous podcast, the parasite primarily infects the finger webs, feet, buttocks, and genitals. The signs, small papules on the webs of his fingers, brown lines, and blood clots point to scabies. Weather in Boston, 62F. It seems true that fall has finally just begun.
Right.
And Daniel.
Zachary writes, Greetings, TWiP. I just recently discovered your holy trinity of podcasts and have been burning through episodes like crazy. My favorite of the three is definitely TWiP. The rapport that you three share is very endearing, and it is quite evident that you all enjoy getting together and discussing such neat topics. I am a lab supervisor at a small rural clinic on an Indian reservation in eastern Washington state, with a background as a laboratory technician in the United States Army. My background is primarily in blood banking, and at one point I was in charge of our Look Back program and responsible for notifying our donors of any abnormal test results and getting them referred to the infectious disease department if necessary. My first episode of TWiP was episode 93, and I was thrilled to know the diagnosis right off the bat when the patient said he had been deferred from donating blood due to a positive test. The blood donor screening test for T. cruzi is highly sensitive, and I've had to refer numerous donors for further evaluation due to positive tests. Now, on to my guess. This gentleman and new father has likely contracted the dreaded 7-year itch, otherwise known as scabies, which is caused by the tiny mite Sarcoptes scabii. This mite belongs to the Arachnida class and is usually less than 0.5 millimeter in length. I've had the pleasure to see this tiny devil under the microscope from a patient of ours who had a skin scraping from the webbing of their fingers. Under direct observation using phase contrast, it looks like a globular lump with, you guessed it, 8 tiny appendages. This gentleman could have contracted this mite from infested sheets. However, skin-to-skin contact is the more common route of infection. I should mention that my co-hosts are all scratching themselves at the moment. And I would suspect that he may have been mixing pleasure in with these business trips. As Daniel said on the podcast, though, the treatment would not be any different, and the man should enjoy the birth of his son, daughter, without having to answer questions about his alleged infidelity. The treatment would be head-to-toe topical permethrin for all in the house, and a thorough washing of all clothing and linens in hot water and dried with hot air. I will be burning through your back catalog, but look forward to the latest episode. Take care.
All right. I believe you're next, Dickson Scott.
At the very bottom there, Dickson Scott writes. Let me scroll it up for you.
Please?
There you go.
Keep scrolling. All right. Scott writes, I just want to say that I love TWiP and look forward to every new episode. I think the addition of Dr. Griffin and the case studies was a brilliant idea and provides some great banter. I don't have a background in biology, but I love everything science-related, and I wanted to let you know that you have an audience outside the biology/medical circle. I work as a hydrogeologist for an environmental consulting firm in Cincinnati, Ohio, dealing mostly with active remediation of Superfund sites. And they probably have a lot of them, right? Currently in Cincinnati, it's a pleasant 65F with light cloud cover. I don't usually send in guesses for the case studies, but I felt like I could take a stab at this one, considering the discussion earlier in the show. I think he has a case of scabies. I did some Googling on scabies, and I can see why asking around his sexual escapades may provide good information, as several places list sexual contact as one or more of the most common methods of transmission. I also came across cases of Norwegian scabies, which can occur in patients with compromised immune systems. I was interested to know why the body reacts to the mites this way. I guess I've got more Googlings to do on my lunch break. All the best, Scott.
And then we have one more from Jimmy, who writes scabies.
Short but sweet.
Okay, Daniel, what else happened here?
Out with it.
Okay, so everybody is right. I don't think we had anyone who was wrong this week, which is great.
No.
As opposed to the previous week where everyone was wrong.
Everyone was wrong. So we're alternating back and forth. Now, this week I'll have a case where everyone gets it wrong, I guess.
Even Dickson?
Dickson will get this. Well, okay. Let's finish this. So it really is a classic presentation, and I think that's why everyone got it right. These sort of long brown lines with the dot. But I like to do, and I think this is important, even when you're 99% sure that you know what's going on, rather than applying statistics, you should do a diagnostic test. I used to love making this diagnosis during medical school, and I'll explain why in a moment. But it is a very easy diagnosis to make. You can do a scraping, and you can actually scrape out these little buggers from their tracks, from their burrows, and you can look at them, as I think one of our email writers mentioned. You could see them under the microscope. And what's neat is to see them. It's actually, it's fun to really clinch the diagnosis and know for certain this is what it is, and then you go ahead and treat it. But the other was that when I was in medical school, this is back in the days when you worked way too many hours, and we had a section at Bellevue. It was a dermatological complaints unit, and people would come in, and you would just see patient after patient after patient. But if you made a diagnosis of scabies and you showed that this really is what it was, they would shut your room and you had the rest of the day off.
Which must have been a common diagnosis, because
you wanted to make it as early in the day as possible.
Exactly.
After 4 or 5 o'clock, it lost the
right.
So did you look under the scope in this case?
So unfortunately, we're in the modern world, so it was sent off, and some lab technician somewhere got to have that joy.
They usually use a little sodium hydroxide to dissolve away the surrounding tissue to reveal the mite itself, because the cuticle of the mite is not susceptible to that. So it's another way of identifying. And it lives in the epidermis, by the way. It doesn't even burrow into the dermis.
Dickson, do you drip the sodium hydroxide?
No. You take some scrapings and you put a drop on a slide.
Yeah, we would use a scalpel and scrape this onto a slide, and then you take the slide.
The picture in our book, I actually took that picture under a phase microscope, and they're beautiful organisms, basically. They've got wonderful symmetries, and they're fun to look at. But when you realize what they're doing, of course, that takes all the fun out of it. By the way, they're all specific for each species, too. So if you have a dog with mange, which is a form of scabies, you're not susceptible to it. Nor if the dog shakes your hand and you happen to have scabies, you will not give it to your pets. It's a human to human or an animal to animal, but there's no zoonotic transmissions of these things. At least that's how I learned it from our medical entomologist, Roger Williams, who taught us a lot of our medical entomology here.
Yeah, so maybe we should talk about how did this man get this on his hands? And as I'm reading the emails, I noticed that Dickson is cringing a little. And I'll say, if you go to the CDC, they actually put scabies under the sexually transmitted diseases. But that does not mean that's the only or even the most common way of getting it, particularly on the webs of the hands. That's, if it's just on the webs of the hands, I'm not really thinking that he got it that way. There are other cases where maybe you have it in other locations where I would actually suspect that it might be contracted through sexual contact.
So if it's in the genital area, right?
In the genital area, you think, most of the time rather than getting this from an inanimate source, it's usually coming from direct physical contact with someone who has it.
That's right.
We'll say the fomite, the transmission from an inanimate object, is going to be an issue in a subtype of scabies called Norwegian scabies. But your general scabies is actually be direct contact. So obviously his hands were most likely touching somebody else who had this somewhere on their body, and actually potentially on their hands as well.
Sure.
And if this gentleman continued to shake hands, he could have transmitted it to someone else, because that tends to be this greeting that we all do. You hear someone you barely know and they touch you with their hands, which may be scabies infested, and you squeeze nice and firmly. And
call this the politician's worst enemy.
I can see this being a problem for politicians.
During election years particularly. What I've learned that a very common way of this being transmitted is from pediatrician to his or her small patient shaking their hands. Small children, usually refugee children that have been in internment camps, during the wars, or orphanages that are poorly managed, and there are tons of those out there, of course. It used to be very common for Korean orphans to be adopted by American couples, and many of them brought scabies with them, and to catch it, all you'd have to do is shake their hand, basically, and you've got it. So, I'm really, although I'm sure sexual contact might be one way of getting it, but we were taught by Dr. Williams, maybe he was a bit of a prude, that it didn't attach to many other places in your body besides the webbings on your hands, unless you were immunocompromised.
Yeah, he may have been a prude. Because I've had the pleasure, shall I say?
Yeah, right.
Of seeing it in other places.
You have plenty of experience at this level.
And so, in certain areas where it's endemic, you see it. And it is, I think, sort of looking at the email ideas, it's not very common that you're going to get this from the bed sheets. That's more of a bed bug type thing. And which that makes this quite different from bed bugs, which is something people sometimes worry about when they come in with a rash like this.
Sure. Or body lice. Body lice too. Not head lice, body lice.
But you didn't try and pursue anything with him, right?
I don't think as we got to, I don't think we really need to. What we needed to do is confirm the diagnosis that this gentleman has scabies, and then how do we treat him?
Mm-hmm.
So we understand how he probably contracted and how we treat him.
You have a lot of options.
And I was actually going back to, I think it was Christine who gave us kind of a list of different options, and what I'm going to say is. We tend to use, this permethrin cream, right? This 5% permethrin cream. And why do we use that instead of lindane?
You mean pyrethrum?
Yeah. No, actually permethrin. Not P-E-R, permethrin.
What
permethrin
is that?
Cream.
What is that?
It's actually an extract from a flower, is my understanding.
Oh, it comes from chrysanthemums.
Is it? Yeah, it's from chrysanthemums.
Okay. That's pyrethrum.
Permethrin.
I'm just gonna have Dickson pronounce everything from now on.
Pyrethrum is directly from the chrysanthemum flower.
Permethrin.
Yes.
Is the name of a drug. It's used to treat scabies.
Elebite.
Yeah, but this is not the drug name. This is the generic. This is not the generic name. Rather, it's
a trade name.
It's a trade name. Yeah. But pyrethrum is the compound. I believe.
And it is a pyrethroid compound.
Yeah. And it's rapidly degraded by sunlight, and that's why it's used. Because it doesn't act as a residual.
Well, it's phenoxybenzyl dichlorophenyl dimethylcyclopropane carboxylate.
Well, I would have said that, of course, but it just doesn't roll off my tongue like it does yours.
It is a pyrethroid. Okay,
yes.
Got it.
Yeah.
It's also used as an insecticide or as an insect repellent.
In fact, that's how the plant uses it for. That's why they discovered it, because pyrethrum containing plants aren't usually attacked by lots of insects, and so just looking around for those plants would identify them.
So this is what you gave him?
Yeah, we used to use lindane, right? Quell was this,
that's a tougher
but
one.
there's a neurotoxicity,
That's
and
right.
so
That's right.
in general in the U.S. we've switched over. But another option is good old ivermectin.
You betcha.
But they've done head-to-head trials, so the ivermectin can work, a single dose,
orally.
Single dose, but the Elamite, we're just going to use it simple, the Elamite topical cream that we treated him with actually has higher efficacy. Sometimes you need to retreat.
Could you use ivermectin in a cream containing dimethyl sulfide? sulfoxide, DMSO, and get it to penetrate into the webbing of the fingers?
Yeah, I mean, we tend not to do that, but I don't see why it...
Because, they used to do that with thiabendazole for creeping eruption, which we've had on this show already.
Yes, we did.
So did you give him any counsel about his wife and their home?
We didn't treat the wife or the child. We did talk to him about the fact that this was contagious, and so he should be treated right away before having contact with the child, and that they should keep an eye on the wife and the child, and if there's any concern, then they could be treated. Again, not with ivermectin, not with lindane, but with this topical preparation that I can't pronounce, apparently. I'm gonna go with the brand name. I'm going with Elamine. But
pyrethrum.
And that was it. You never heard from him again. Right?
Then it was all good. Okay. Everybody was happy.
The other thing is that these are little tiny critters, and if they don't find a host soon, they dry out. So they might have a half-life of about a day and a half or something like this, but it's not longer than that. So if a lot of the treatments for these infestations, so to speak, and there are some bird mites that will cause problems too. That I said these are specific for their hosts, but there are some that jump the borders, and Dermanisis mites are the ones that can do that. They recommended just turning the heat up in the house and leaving it for a day to increase the temperature and decrease the humidity, and it dries out everything. And of course, these things would die under those conditions. So
yeah, they really need people. They really need a person to live on. And there is, I guess, the other form we should discuss, which is the Norwegian scabies.
Well, yeah, that's an extreme situation,
which is really a very different manifestation. It's people tend to have these really thick crusts, and they are just teeming, I'll say, with scabies, and highly infectious.
That's right. But they should change the name, because I think that's ethnically profiling. It's not just among Norwegians.
Let's call it Norwalk.
Well, it was described originally.
No, I appreciate that, but I'm sure the Spanish flu is not appreciated in Spain any more than the Norwegian scabies is appreciated in Norway. So, they did change the name of the virus to take that away from.
That's right.
So if you had to call it anything, I would just call it immunocompromised scabies. Hyperscabies.
Hyperscabies. I like hyperscabies.
Hyperscabies.
It's a case of hyperscabies.
Hyperscabies, that's right.
Hyperscabies.
And treatment with ivermectin is the way to do that one.
What a coincidence.
It's amazing. It is. Every now and then the stars do come together in the right order.
All right, let's move on to a paper.
Do that.
Because I know we're time constrained.
I'm sorry to say that. You're absolutely
Dickson needs to leave. The paper, this was actually suggested by Daniel quite a while ago.
It was.
Because I think the lead author, the PI Miguel Soares, was visiting. Is that correct?
I think that's true, actually, yes.
Nice.
Now, is this Miguel Soares, was he someone who was here before, do you know? Or so he was giving a seminar, right?
He was giving a seminar, and I actually ran into him before the seminar, and we started talking.
Did you know him, or you didn't know him?
We sort of had shared, I guess, research interests, because I did a lot of work on something called a B1 cell, an innate B cell. Mm-hmm. And so he was familiar with that. And it comes up a lot in the idea of where do we get this protective shield of antibodies. So I was familiar with his work, he with mine, and when we got to talking...
So the title of the paper is Gut Microbiota Elicits a Protective Immune Response Against Malaria Transmission.
How can that be?
This was published in Cell. Well, we're going to find out, Dickson.
Well, that's what everyone out there should have said. No, come on now. The gut microbiome can be responsible for everything.
So Jonathan Eisen, who is a microbiologist at UC Davis, likes to call these things, it's not necessarily this, but... This week in microbiome overstatement.
It's probably right.
Because the press often overstates the value of the microbiome. But in this case, actually, this is quite good.
May I interrupt
for of course.
One moment? There is a huge, successful exhibit of the human microbiome at the American Museum of Natural History as we speak. Highly recommend.
Have you been?
If you're in New York City, not yet, but I've seen the previews because I get their magazine because I'm a member of the magazine.
It's in New York, huh?
It's in New York, and it's featured in the, I think,
Biodiversity,
Hall of Biodiversity.
When they were developing that, I was invited to go down and look behind the scenes.
And you didn't.
No.
Of course not.
I can't do everything. I have to
well, that's my answer, too.
I have to parcel my time.
But I'd love to go maybe the three of us should go and have lunch at their cafeteria. And then
Why does
visit
it
the
always
microbiome.
involve food, Dickson?
Well, because it's got microbiomes. Come on.
Why don't we
gotta
just
feed
go
them
see the
microbes.
exhibit?
Yeah, we could spend time. Yeah, I agree. I'm up for that. Let's see the exhibit.
The best thing would
You
be,
can eat first.
The best thing would be if for our 100th we could do one right there, but they won't let us do
Oh,
that.
I bet you they would.
Although the woman
I
who
bet you they would.
invited me to go behind the scenes, maybe I could ask her. We could just sit in front of an exhibit and do a.
The current president of the American Society for Parasitologists is the parasitologist at the museum.
But we don't need to do it during hours. We could
No.
do it after hours.
we could do that.
Then it
after
would be
hours.
at night at the museum. Right?
Exactly right. We could invite what's his face,
What's his face?
that actor.
All right. So this is very interesting because a while ago I wrote a blog post called "Natural Antibody Protects Against Virus Infection." We have something in us called natural antibody,
Yes.
which is directed against the disaccharide galactose alpha-1,3 galactose, two galactoses joined together, which is a terminal sugar on glycoproteins. Humans, apes, and old world monkeys lack the gene that encodes the enzyme galactosyltransferase, which attaches this to membrane proteins. And so we do not make alpha-gal.
We do not.
We do not.
Thank goodness.
Therefore, our gut bacteria do.
This
So
is true.
we have in us over 2% of our serum antibodies are against alpha-gal
It's incredible.
in us, but that must vary depending on where you are, as we will see in this paper. And so this is it's called natural antibody because it's thought that if you get infected with something that has alpha-gal on its surface, then these antibodies they're already there. You don't have to wait 2 weeks for an immune response.
Right.
And it will combine with complement to bang
about
whatever's
that?
coming in.
How about
And an
that?
example, envelope viruses that come from animals that make alpha-gal transferase.
Yes.
They will have alpha-gal in the envelopes of these viruses, and as soon as they come in, bam, they're gone. And this is thought to be the reason why we often are not infected with enveloped viruses from animals.
Interesting.
Now, obviously, this happens now and then because Ebola comes from an animal into a person, but maybe that
Yeah.
person had low alpha-gal and antibodies in their
Maybe,
serum, right?
maybe.
So that's the background here. I think it's quite interesting.
Yes.
And basically, what they want to show or look at in this paper, whether this is of any consequence for malaria.
Right. Because
Go ahead.
that's not out of the blue.
It's out of the green.
Daniel
green
can
screen.
say, well, yeah, it's out of
Actually,
the
yeah, the
fluorescent
bigger
green
one,
protein.
it's out of the fluorescent green. Because the malaria sporozoites
Yes.
have alpha-gal on their
Dixon,
on the
what's a
surface.
sporozoite?
A sporozoite is the infectious stage of Plasmodium.
Does that come? I thought the mosquito gives you sporozoites, right?
This is correct.
Yeah, out of its salivary glands.
This is exactly
All right.
correct.
So this has been shown by others to have alpha-gal on its surface.
How remarkable. Because it also is completely covered with another protein called the circumsporozoite protein, which is a quadrapeptide.
That must come from the mosquito,
What must?
the alpha-gal,
Maybe.
because people don't make it.
Maybe. Bingo.
It has to, right?
It could.
Because we don't make alpha-gal.
It very well could be.
Yeah, because it doesn't. These mosquitoes don't infect any other animal but humans. Well, they don't get their parasites from. What's the form that it picks up from an infected human?
It gets the gametocytes.
Gametocytes. It doesn't pick up gametocytes from some other animal.
No, but what you're implying is that the metabolism of the malaria parasite doesn't make alpha-gal?
Maybe it does. Yeah.
It could. I think it could.
Should we give a
It
little
must be
background
it.
on what
Must be it.
alpha-gal is?
We should.
We
What
just
we're
did.
talking about?
We just
we
did.
did? We feel like we got it? You
not
feel like
just-
your background was good?
It's not just any
disaccharide.
other woman.
Unless you want
It's
to
an
add
alpha-gal.
unless you want to add something in addition, go right ahead.
No, I think
But on
of-
TWiP, I'll say something, and then 5 minutes later, Rich Condit repeats it. And it's
Does-
my like,
does-
where were you? Did you go to the bathroom?
I think he
No,
does.
but the
I've listened
electrons
to-
don't travel that fast down there. I don't know.
That's
something
all right. What
about
else
that.
would you like to
Well,
know?
I think we normally think of proteins as what the immune system might be targeting, and what this is it's a certain type of sugar that decorates the proteins.
Yeah.
And there's an enzyme that basically creates this type of linkage, this sugar.
Right.
It's
It's a
antigen.
glycoprotein.
Exactly.
Yes, it must come from the Plasmodium.
I think it probably
sure,
does. I think it probably
absolutely.
does.
So, in fact, the first experiment they do is to ask, do sporozoites have alpha-gal? And they
Yes.
do. They do some immunofluorescence, and they show you can remove it by an enzyme. Dickson, what would be the name of the enzyme that removes alpha-gal from protein? What would you call that?
I would call that, let's see, a galactosidase.
Very good. Yeah.
Alpha-digalactosidase.
That's the beauty of, it doesn't have to be di, it could be just galactic. It's the beauty of naming. You can tell what things are. Yeah. That's why taxonomy is great, and people are bored with it. But
one of my favorite
it's not
words of all time, since I spent some time with Christian Deduve's laboratory over at Rockefeller, because they worked on lysosomes, was N-acetylglucosaminidase. I loved to say that. And then I said, gee, what does that mean? But it's one of the enzymes that is important for microbial defense.
N-acetylglucosamine is a wonderful word, but the abbreviation is NAG. And I think that's not beautiful.
Nagase.
Nagase.
Right.
Then they find that they do this correlative study in kids in Mali, where apparently there's a lot of malaria.
Right.
330.
Lots of it.
Plus 229 kids. And basically they find that in kids less than 2 years old, the average level of anti-alpha-gal IgM is 33 micrograms per mil. And it's much higher in adults.
Right.
And so, kids is this correct? Kids often get infected?
Well, there is an age group where it's like 6 months to 5 years. That's the age group that is the most susceptible to the lethal form of Plasmodium falciparum, because maternal antibodies have faded, and their ability to respond to a full-blown infection is less than an adult.
Okay.
So they've got that window where that's where the infant mortality rate's the highest in certain parts of Africa. Would you?
I would totally agree. And what we're seeing here in this figure is that 0 to 1 you have these low levels, but then it rises pretty rapidly. By 2 to 3 years of age, you're almost at this adult level.
Probably because you've had certain exposures to the parasite by then.
Or they're going to claim certain exposures to the appropriate protective gut
or
microbiome.
You've had diarrheal diseases caused by alpha-gal producing E. coli, right?
Well, no, I wouldn't say diarrheal diseases, no.
Well, children do get a lot of them, though.
Yeah, but I think...
Yeah, it's not clear that the bacteria in your gut need to make you sick to educate your immune system.
I think this is part of our microbiome, and these kids are acquiring. Remember the paper we did on TWiP a while ago showing how the microbiome evolves after birth towards 2 years of age?
It's been hard for me to accept that, but I did. I've finally come around to the light force rather than the dark force.
Oh my God. Are you going to stop accepting science at some point in general?
You have to accept the data. It's not a belief system. You've got to accept the data. So
they actually followed these kids through the malaria season and found that the ones who had higher alpha-gal antibodies were less likely to get sick. So there's a correlation. Right. Doesn't prove anything, right, Dickson?
It doesn't prove anything, but it also says that it doesn't eliminate totally the infection. What it does, it modifies the disease.
Oh, no, but Dickson, this is one parameter they're correlating. It could be many other things that they haven't even looked at,
like
right?
nutrition.
So you have to follow it up with other experiments.
You do.
And it's important that it's just the IgM that correlates. The IgG levels were pretty much the same there in this part of Africa as they are in the U.S. There wasn't much of an association between whether or not you got Plasmodium or not. It was just the IgM that was much higher and that correlated with whether or not you got the
So I've got a question for you here. I was confused by this because I'm an old school person that got my education through Elvin Cabot. Elvin Cabot was one of our most favorite. He was one of our most famous.
Really? You did?
Stop it. If you wanted to look like Elvin Cabot, you're going to have to ask me to do that. No, he talked just like that. But he was famous.
We have to get him on TWiP.
Yeah, well, good luck on that. So he was famous for having injected himself with dextrans.
Yeah.
And then measuring the anti-dextran antibodies. And his claim was, and it might still be true, was that, look at him. Look at Daniel's cringing over here. Do you know how many doctors gave themselves diseases to see what they were like? Plenty.
I think there's a Nobel Prize winner, right?
There's a couple of them.
A couple of years ago who had done that, but apparently it's still done.
The point is
the
that...
H. pylori guy?
Yes, exactly. That's what I was thinking of.
So he was an Aussie, right? So the antibody, the class of antibodies that he claimed persisted after polysaccharide exposures, but not protein exposures, was the IgM class. You go along with that?
Well, persisted, not just were there initially.
Yeah, it persisted, and it was. But I'm confused then with repeated exposures to the same antigenic epitope. Why don't you switch to an IgG class?
Well, typically you do, right?
Well,
I don't know. But you're kind of being an anecdote here, Dickson, so we don't know.
No. I'm asking an
you remember
immunologic
this
question.
You remember this is what he told you, but maybe you don't remember it quite well because the mists of time are clouding things.
It's true. It's like the shadow clouding men's minds. But the point is that they see higher IgM levels.
I know, but later we're going to see that it can also go with IgG. So don't get too hung up on it.
Repeated exposure should have switched everybody over to an IgG response.
But the kids don't have all that much exposure, right? They have 1 or 2 exposures early on. But also, it's not exposure to the parasite probably. That's
just the
doing
epitopes.
This. I think it's the bacteria. As we're going to see, it's probably the gut bacteria that's doing this.
Yeah,
so we think anyway.
No, I actually think what Dickson is raising here feeds into our understanding of who's making these. Who's making these antibodies?
Right.
And we break down...
What do you mean? Who's making the antibodies?
Which cells?
Which type of B cells are making these protective antibodies? And this really makes the distinction between your adaptive B cells and your innate B cells.
The B1 cells might be active.
Well, the
here?
B1, and actually, I'm gonna actually admit here that I think these are made by a group of innate B cells, the marginal zone ones. I don't think these are B1 cells, but B1 cells and marginal zone cells have this ability to create a level of protective IgM that won't necessarily go on to make an IgG response. Your classic B2 cells, you get your injection for tetanus. First you see IgM, then it goes to IgG.
That's right.
And that's sort of your classic. But your B1 cells, some of your marginal zone cells...
Are these pirate patches and lamina propria cells?
There's always been a little bit of a controversy of where do the B1 cells and the marginal cells go, right? So the marginal cells are going to be in the margins of a follicle. The B1 cells are a lot of, I'll say, serosal surfaces basically in the gut.
Right.
And so
Mucous membranes.
Do they really have to go to Pyrus patches to create a low level of secretion? No, but the high level secretion, they probably do go to the spleen or to a lymph node or to a Pyrus patch.
So these don't require somatic hypermutation then because they don't go to IgG, right?
These cells actually can produce mutated IgM. They tend to be a lot less mutated, but they can do that without necessarily isotype switching to an IgG.
Because they show later that AID is not required for protection, that's the enzyme involved in somatic hypermutation.
Yeah, most of the coding of this is evolutionarily selected and in the genome and doesn't require a mutational affinity maturation.
Remarkable.
Right. Interesting. All right, so mice have the gene for this enzyme, alpha-1,3 galactose transferase, and they have made mice that are deficient in it, which people were already deficient in the gene that makes alpha-gal. And they found that if they had pathogen-free mice, they could give them, expose them to E. coli, a specific strain called O86B7, and this would induce antibodies to alpha-gal in these mice. And it would protect them from infection. They have an infection model with mosquitoes and immunizing them basically with E. coli or giving them E. coli orally induces antibodies to alpha-gal and protects them from a transmission with infected mosquitoes. They use Anopheles stevensii. Stevensii?
Yes.
Stevensii. I'm making the Italian version. The Italian version.
It's okay.
So why they picked E. coli O86B7?
Must have had a lot of alpha-gal.
Is a good
question. I think that's why they picked it. It's a strain that does express a lot of alpha-gal.
Yep.
I happen to know that the O refers to the O antigen of lipopolysaccharide, like O157.
Right, which is very dangerous.
And O157H7 is the one that can cause
that's a
gastroenterizing, and you get it from undercooked hamburger meat.
That's a jack-in-the-box.
The H7 is a flagellar antigen, but I don't know what B7 is.
No,
maybe it's a typo, but they typoed it over and over.
But notice that they had to use the complete life cycle in order to get this to work. If you had just transferred this through transfusion, I would have been very interested to see what happened
in the mice.
Yeah, or even in humans. I mean, the sporozoite is the stage that has a lot of alpha-gal on it, so it's the target for the IgM antibody. Yeah. But what about the other stages of the parasite, which are intracellular? and which then replicate and then briefly go into the serum before they go into another cell.
They do an experiment later where they inject it. We'll see the result of that. Excuse me. But this, interestingly, they feed these mice this 086 strain, but they first treat them with antibiotics to deplete their gut bacteria. So otherwise, I think this thing wouldn't take.
Right? I think that's the challenge.
And so you can induce protection by giving the mice this particular E. coli. And then they do a very nice bunch of experiments where they show that they use a variety of double knockouts which lack B cells, or they lack IgM, or they fail to undergo class switch recombination or somatic hypermutation. It's wonderful that you can do all this stuff. Yeah. So basically they show that the protective effect of the E. coli is via a mechanism mediated by anti-alpha-gal IgM antibodies that do not undergo somatic hypermutation.
How about that?
So they can discern that from all of those. Isn't this wonderful? This is beautiful.
With some interesting graphics.
It's beautiful work. It's beautiful work.
Yeah,
really
they're
nice.
very interesting graphics also, I think.
Why do you say that, Dickson?
Because I'm not used to seeing data expressed this way, because I'm old school. Perhaps
You're old school.
But we do have...
He keeps mentioning
we
that.
do have,
They haven't read papers in a long time,
we
right?
do have... Yes, we have. Stop it. No, there is a graph here on figure 2, which Daniel and I both looked at and said for B that maybe those two groups were not necessarily significantly different because the error bars overlapped so much with each group, but the statistical method that they applied was not widely well known.
Was it parametric or non-parametric?
It was non-parametric.
No, I'm pretty sure this was parametric, actually.
You mean a pair of metrics did this?
You know what they call this kind of plot, Dickson? That's called a whisker plot.
That's very interesting to know, Vincent. That doesn't help me understand why I can't see the big difference between these two groups. Because they don't look like much different to me.
Well, we can't really go into figures because our listeners don't have this figure in front of them.
No, they don't.
We have to explain it for them.
We do.
And now they take these mice, and remember, they've put E. coli in them, and they're protected against Plasmodium. Then they just immunize them with alpha-gal.
Right.
Using rabbit red blood cell membranes that have a lot of alpha-gal in them, and it makes antibodies in these animals, both IgM and IgG, and it protects them against infection. Again, so you can immunize them with an alpha-gal containing vaccine, and it will protect the mice. And again, they're delivered by mosquitoes.
Yes.
Stevensi or what?
Stevensi.
Why can't I remember?
I don't know. we complement each other very well here.
Now
I'll pronounce and you think.
Protection from artificial transmission via intradermal inoculation, but not when sporozoites were inoculated intravenously.
Yeah.
So they say the protection is somehow in the dermis. What do you think of that, Dickson?
What do I think of that? I don't think that's the reason. I think the real reason that intradermal injection, mosquitoes don't inject intradermally.
What do?
They suck blood.
They don't go with their, what is it called? They
injected intravenously also.
The proboscis.
Proboscis.
The hypostyle. It's the hypostyle.
You always say hypostyle.
Well, that's what it is.
Does it not go into the dermis?
It goes through the dermis and it penetrates into a capillary. That's where they get.
Are you sure?
Where do they get their blood from, Vincent?
It penetrates a capillary.
Where do they get their blood from,
Do they have to do multiple soundings? They go right to a capillary? Is the capillary bed that dense?
Have you ever seen a woodpecker look in a tree for a grub?
How much wood could a wood
That's a woodchuck. That's a woodchuck. They can detect them by vibrations. I'm sure that this mosquito detects it by the heat difference.
Oh, really?
They have
Daniel, think of the capillary bed in the subdermal.
But think of the size of a mosquito.
Is it that dense that a mosquito would just hit it?
It's not the density. It's actually something about the incredible ability of the mosquito.
Really?
And it's probably as heat. I'll say it's probably heat directed.
It is heat.
There's slight differences in the temperature, and they will aim and they will actually insert into. Basically, they've got we use ultrasound nowadays.
Oh my
they use a heat-based censoring to actually put the hypocell right into the capillary.
This is
intravascular.
This is a tiny thing that lives for 2 weeks.
It's true.
And that's it. And it's incredible.
Look at it.
They're like disposable needles.
So
flying disposable needles.
They've called it. So imagine if you injected the sporozoites intradermally.
Yeah.
That's not what a mosquito does. They inject them intravenously into the capillaries.
Well, then this result is spurious.
No, this is actually what they're trying to make the point of, is that the antibody response that protects us does not come from necessarily from exposure to the sporozoites. It actually comes from this communication, this education that our gut microbiome is giving to these kids.
So the antibodies against alpha-gal are not hitting the sporozoites?
No, I don't think the sporozoites succeed if they're injected intradermally. I don't think they complete the infection.
No, in control mice, it did complete infection.
Intradermally?
Yeah.
Well, then they must take their time getting into the bloodstream then.
They just say that once the sporozoites are in the blood, the anti-alpha-gal antibody-driven mechanism is no longer effective.
Yeah, but if that's true, and if mosquitoes inject the sporozoites directly into the bloodstream, then how does
I agree with you. That's why I say the
that
results
becomes a challenge.
How does this work?
So that's why I...
Excuse me, because, I want to tell you something. When the mosquito injects the sporozoites into your bloodstream, it's a matter of minutes before they get into the hepatocytes.
Right.
Minutes.
30 minutes.
Less than that, probably.
Mm-hmm.
You only need one to generate the whole infection.
Then it gets into a liver cell.
That's
and
correct.
At that point, antibodies...
It's got to go through the sinusoid first, so there's a receptor for that. And maybe that's what the circumsporozoite antigen really performs. It's a receptor, just like viruses have receptors, and I know you know all about that. This sporozoite has a receptor, and it's on the surface of the endothelial cells that comprise the sinusoids of the liver.
Mm-hmm.
And then once it's through the sinusoids, it now has to go through it's now into the hepatocytes, and it can get into an hepatocyte.
So
but it's quick. It's very quick.
I'm puzzled by this result.
Me too, because it's a matter of time of exposure. Is the sporozoite exposed for a longer period of time if they're injected intradermally than if they're injected intravenously? And I would say that, yes, that's probably true. Then they can be damaged.
Well, remember, in their mouse model, they're having mosquitoes bite them, and they're protected by having anti-alpha-gal antibodies. So there's a little disconnect between that and their injection.
There's some disconnect here.
Actually, I'm glad they do it with the mosquito bite, because it really recapitulates what
That's
happens
the
in
whole
nature.
Schmear. That's right.
Fortunately.
That's right.
Well, they go on to show that in their mouse model, IgM is enough. IgM anti-alpha-gal is enough to protect, but it's better when IgG are present.
Which is a much more efficient or avid.
And IgG2B
it's more
in
avid
particular.
than IgM?
Well, IgG can fix complement, right? IgM does not, right?
That is true.
But so maybe that's the killing mechanism.
I think they show that complement is activated and is involved in the killing, possibly in lysis of sporozoites.
That makes sense. That makes good sense.
But what IgM is doing is not clear. They say there are... What do they say? They do say something here, which I had underlined and meant to tell you about IgM. IgM has no... let's see, it's in the discussion. The protective effect by IgM antibodies... that's what's important in people, right?
It's a pentavalent antibody, right?
Yes. I can't find
But the avidity of IgM is low. I think the binding constants are low. That's what I'm driving at.
You know what they say, Dickson? The protective effect of anti-alpha-gal IgM might contribute to explain why only a small fraction of sporozoites inoculated by mosquitoes manage to progress towards establishment of infection.
It's fair. You only need one to succeed. That's the point. It doesn't matter if it kills off 99.9%. If one succeeds, you're still going to get clinical malaria.
So they're saying maybe we can immunize people and give them antibodies to alpha, or boost their anti-alpha-gal antibodies. Maybe that would help.
Maybe
that would be easier than making a Plasmodium vaccine, as you know, which is really
hasn't happened yet.
Been difficult. A lot easier to get alpha-gal.
It would be very easy.
So, what do you think? Is that a reasonable? Should we do a clinical trial?
All right. Well.
Hard to say. I don't like that.
Let's ask the clinician.
No, I don't like that clinical trial.
You don't want to.
Well, I was going to say a couple of things. I don't want to make a broad generalization about IgM. There's different complement fixing of different subclasses. Not that IgM fails to fixed complement. It just,
Not efficient.
there are other IgG subclasses, which have better complement fixing properties. Right. But the other thing I wanted to say is it's important that before you make your classical vaccine, you think about this distinction that we try to raise between innate B cells and adaptive B2 cells. The B2 cells are classically the ones that respond to our traditional vaccine technology. Yeah. They'll get together with T cells, maybe with a conjugate vaccine. They'll do it straight away with a polysaccharide vaccine. They'll go to a germinal center and they make all these antibodies. But your innate B cells don't respond the same way. And since this is one of these evolutionarily selected antibodies, maybe the way to get them to respond is through this gut education response. 80% of the B cells in the body are in the gut mucosa. So if you really want to educate the students, you go to where the students are. And you go to particularly where the students you're interested in educating are. You're not interested in educating these B2 cells because they don't really seem to be the source of this wonderful protective antibody.
They did immunize the mice, IP, I suppose, with alpha-gal containing antigen. That worked. So
because again, IP, right? They're putting it where the B1 cells are, where the innate cells are.
Well, let me make sure it was IP. Are you sure about that? Because I just threw it out there. Immunization, IP, yeah.
IP.
That's an easy way to do it.
It's very simple.
Would you like to be immunized IP, Dickson?
No. Thank you. No, thank you.
So
orally,
okay,
that's
so that's
the way I want
interesting.
it.
That would be very. So maybe if they just put it in your arm, it wouldn't work because you're not hitting these.
Well,
gut
orally,
cells.
I think orally would be a good way to do this.
Would you like to have alpha-gal orally or this E. coli strain orally?
No,
Probably
give me the alpha-gal.
Yeah.
I'll take the alpha-gal.
So that we should say that they believe that as we are colonized with our microbiome from birth onwards, E. coli producing alpha-gal or maybe other bacteria are introduced to us, and our levels of alpha-gal slowly go up, and we saw that in the graph, until they reach adult levels.
Yeah, but guess what? Anybody who's never been to a malaria area and then visits a malaria area gets it worse than anybody else.
Yeah.
So we must have lots of anti-alpha-gal, but that still doesn't help us.
It's not the whole thing. There are other antibodies against malaria proteins,
of course,
right?
Of course. But the point is that we end up suffering much more because
absolutely
never have a history of it.
So there's also this interesting allergic issue about this anti-alpha-gal allergy. And it's actually quite prevalent in certain parts of the country. In parts of the country where they have a high prevalence of the Lone Star tick. There's something in the Lone Star tick salivary pharmacopoeia. We don't know what it is exactly, but when it injects this into a person, they end up developing a lot of, and actually IgE antibodies to alpha-gal. And why is that a problem? Why would it be a problem to be hypersensitive to alpha-gal?
It's a homocytotropic antibody.
Because if you eat any animal other than an old world monkey or an ape or a human being, you can develop this, what they call meat allergy. I mean, anything, you can eat fish and poultry and things like that, but, red meat.
But we already have alpha-gal producing bacteria in our gut.
And this low level of IgM doesn't produce this allergy. It's when the B2 cells see it.
So people who have gut inflammatory issues, maybe this is part of that problem,
that potentially
right?
could be part of it.
Very interesting. Yeah, that's right.
So there's certain antibodies that it's okay for the innate cells to make, but when you get the B2 cells making these antibodies, it can get you into trouble.
Go ahead. No, I just spent half of my adult life in the laboratory angsting over the immune mechanisms involved in a worm that has part of its life cycle in the gut tract. And I had a lot of colleagues that the more immunology that was learned about the nuance of cytokine communications and the kinds of B cells that there are, the less likely it was that their original hypotheses were correct. And they had to revise and revise and revise. And I don't know how many times you have to revise until you finally get it right, but I don't think we've got it right yet. So we still need to know more about the gut.
Just think if you were working now, all the techniques.
My mind blows.
It does?
Smoke rings thinking about it.
You have a bad mind?
No. I stop it.
You said your mind blows. That means it might.
I didn't mean to say it quite like that.
In 60s speak, that means you're not very good.
No, it's blows my mind to think about the
okay.
possibilities.
Anyway, I like this paper because
Yeah,
I
raised
know
a lot of
this
issues.
alpha-2 antibodies are protective against viruses from other animals that
So
come
there's
from
There's
animals.
a bridge.
So this is cool that it's parasites as well. And you know, Dickson, are there other parasites with membranes that could have alpha-2 gal in them?
No question about it.
No?
Lots of them.
Leishmania,
Perhaps.
trypanosomes,
Perhaps.
Giardia,
Perhaps. Toxoplasma.
Toxoplasma.
Perhaps.
How about worms?
We'd have to know about the worms. Mostly they're secretions of the antigens in this case, and those might have alpha-gal too. A lot of them have choline, of all things. They have choline that they secrete that alters the serotonergic nervous system. It's kind of a weird stuff.
All right,
Weird
now,
stuff going
Dickson,
on down there.
We have 12 minutes.
We do to present
Let's
a case.
do a case presentation and
Here
then
we go.
can wrap it up.
Here we go.
All right. Now, I have Dickson can look. I don't have anything here that Dickson can cheat off of. No, maybe
He's
I
saying
do.
he would.
Yeah,
I would never,
I'll
ever
put away-
think of doing that.
I'll put away the answer there. I'm going to present. Okay, I'm going to present two cases. These
Two.
two people have the same thing.
Oh.
Okay. And I'm going to... Okay, well, let me tell the story, and then I'll throw in some stuff. So we have a 12-year-old girl and a 5-year-old boy. We'll just start with the 12-year-old girl. This 12-year-old girl initially presented for care with a painless dark brown area on her right knee. She reports that she has brothers and sisters and that they often play together outside the house. She does not report any specific trauma to this area, but she recalls that the area started to expand. She is noted to have this painless dark swelling of the right knee, and then she develops cognitive changes. Okay? And she's going to go and get an MRI that we'll discuss. Now there's this 5-year-old boy. He's from a coastal region. I'm going to mention that both of these patients are from Peru. And this 5-year-old boy is brought in by his mother, and what he has is a swollen area on the face. The mother says that while he was playing, he scratched his nose. It started to swell. It started to spread. He was given some antibiotics, but they didn't help. The swelling extended around the right cheek, around the eye. He started to develop some nasal obstruction, and the whole area was painful. And he also had an MRI. And I'm gonna tell you what the MRIs show. Each MRI is going to show an infiltrating process. The woman, it's going to be in the left side of the brain, in the middle cerebral artery territory. And the boy, it's going to be this infiltrating process in the frontal region, mostly in the midline area. Okay, so we have two patients with a skin lesion and an infiltrating process in the brain. And both of these were seen down in Peru. And so before you ask questions, I'll put in a little plug. I saw both of these patients when I was down in Lima recently. And I was at the Gorgas Institute down there. And a fantastic place. Not only do they provide great care for people, but they provide a lot of free medicines. So, if people want to donate to them, please go ahead. But let me say, what questions do people have?
So are they sexually active?
5 and 12, even in Peru.
So neither of them
Don't
are
think so.
No, neither of them are sexually active.
That was just a silly thing. But
Yes.
I always ask about that. They haven't had any changes in diet.
No changes in diet.
How long? I'm sorry.
Go ahead, Dickson.
How long after they noticed the lesion did they come into the clinic?
Months. Many months. Months.
So how could they think back that far and associate? Well,
Are these lesions gone at the time they come to the clinic?
No, they come to the clinic.
still there.
And they both have pretty significant changes in the skin. Now, the boy's is more of a red, swollen, and the woman's is more of a brownish.
So it took
And
months for them.
Neither of them hurt. Well, it didn't go away. It didn't get better. The boy,
but they had cognitive symptoms.
Well, the woman was prompted to get an MRI because she had cognitive changes. The boy, because it was in the face, and they wanted to get better imaging about, is this going deep? What is this involving?
And indeed it did in
And
both cases.
both cases,
How long
they saw
after
brain
the
involvement.
knee the knee changes, did the cognitive changes come on?
Many, many months.
Many months.
Wow.
Wow.
How do we know that these two things are related?
I was also going to say that too, but you don't. You obviously
do not.
Yeah. When we see each of these cases independently, we're not sure what it is. But then when we get the diagnosis, we realize each of them have the same diagnosis. But it's this combination that should be making us think of a long-term, non-resolving skin lesion and infiltration of the brain. It's not a meningitis, it's actually infiltration of the brain itself.
Is there, when you look at the MRI, are you allowed to describe more characteristics, or is that going to give it away, so to speak?
No, I can even show it to you, but...
is it a space-filling lesion, or is it a solid mass, or is it?
It's infiltrating. There's a whole area that appears to be infiltrated with some sort of a process. So it's not a separate tumor. It's not a mass.
Denser than the surrounding brain tissue, then.
Mm-hmm. And there is edema there.
So
and there is edema.
Can I ask some more
Vincent, go ahead. Yeah.
So have they been swimming lately, or been near any sources of water?
No, I think that's an excellent question. They don't describe any swimming.
And they haven't been playing in the water. Where they've been playing is dry.
Well, it's I wouldn't say it's dry. Sometimes it rains. It's muddy at times.
And where do they live? What kind of home are they in?
Pretty rural. So they're playing in dirt areas.
All right. They don't have ducts and air conditioning in their homes, right?
No air conditioning. No screens on the windows.
And is this a wooded area, forested area, or is it just a plain and dry?
It's forested.
Oh, so they play
sort
in the woods.
Jungle
they
type
play in the
area.
woods
Yeah,
as
they're
well.
playing in the woods.
Okay. And they play quite often. There's no water around, though. There's no like standing.
There
pools
wasn't
of
like
water.
a standing pool where
But
they
were
had been
playing.
swimming or.
Okay, so they have
diving.
no report of being in contact with water, but they are in contact with dirt and wooded stuff, right? Okay.
Interesting for this to smolder so long, though. No matter what. That's my confounding.
That's confounding to you? Yeah.
To me it is, yeah, a little bit.
All right.
But I've still got my ideas.
And they're young, so they don't have, let's say, neither of them have other medical problems going on.
Right.
We don't pick much up as family history. They're fortunately young enough not to have developed any toxic habits, as we say.
Yeah, did you biopsy the lesions where they were, where they scraped themselves?
So that's potentially a diagnostic test that someone might want to suggest.
So the boy scratched his nose, doing what?
Well, that's the story that the mom gives.
Did he
That there was
scratch-
initially a scratch on his nose that he got while playing, and then
by
this
doing
developed.
what? Did someone scratch him, or did he fall and scratch it in the dirt?
He was out playing.
We don't know.
We don't know.
And the girl, same thing, had some kind of trauma to her knee. She fell, or
That's what she reports.
Okay.
Not that she had a major trauma, but that she may have fallen down, she may have scraped her knee, but we're not getting any kind of major trauma that preceded this.
okay. Well, I think right now I need to ask questions that would help our listeners, because I have an idea what this might be, right, Dickson?
I hope you do.
And do you? But
I
you're
do.
puzzled with the long incubation period.
Yeah, but that might just be a quirk of these two cases. It's not usually that long sometimes. The fact it's in the brain.
Sorry, and this was in a city area or a rural?
Both in rural areas.
Both in rural.
They came to Lima for care,
Rural.
but this was acquired outside.
Right.
And they had, you said they had altered cognitive.
So the woman had altered cognition. She didn't seem like she was the same. Something
Okay.
was up, and they
No
did the
nausea,
MRI.
though?
No nausea, no vomiting.
Okay.
And the boy, it was just because it involved his face. They thought we should do some imaging
And you happened to see it. So it was
and you
a,
could see it was involving the brain.
what do they call this? A incidental finding, right? You find it when you're looking at something else?
Actually, they were not looking specifically
Yeah.
for something infiltrating the brain.
Yeah. Okay. Dickson, what other questions would help our listeners with this one? Actually, I think they have all they need.
I do too. I do
But,
too. It's not scabies. Something apparently, okay, so apparently if you connect the dots in this case with the information that you've got, it had to start with a contamination from the soil.
Yeah.
It's slow burning type of lesion in the skin, And then the next thing you know, it was neurologic.
So the lesion developed rather quickly, though, after the injuries, right? Would you say in the first week or so, or longer?
The skin lesions.
I don't know. I don't know if we have that really pinned down, like how well,
All
but
right.
you have made this diagnosis, and
Yes.
therefore you could speak to the general issues as we come to discuss the case.
What would we suspect it to have done?
Yeah, exactly. Exactly. Right now, looking back, hindsight is 20/20.
Yeah.
But right now, we're not worried about hindsight. That's about it. Besides immunologic tests and biopsies and stuff like this.
Yeah,
It's plenty of it.
and I will tell you that, just to give our listeners a little bit of a hint, that we do a biopsy, and that we'll be discussing the results of that biopsy. We do a biopsy actually in each case.
You're not going to take any CSF, though, are you?
We don't, but
Did you take biopsies from the brain and the site of the lesion?
so I didn't.
No, but did anybody? 2 places rather than 1 place?
No, it was actually, when
brain
we saw
biopsy. Brain biopsy,
after the biopsy we made, I won't be specific in where the biopsy occurred. We make the biopsy, and we'll see if whether that or or a serological test or a culture is key in getting this diagnosis.
Yeah, this doesn't. Yeah, that's
So
right.
people can give us suggestions about what we should do next, and they also can take a stab in the light.
No
Stab.
pun intended.
Okay.
At what they think this might be that we're seeing.
Right.
Okay.
And I'll say this is not a very common diagnosis, but it's interesting that I saw a couple of them while just in Peru recently.
So have you, is this the first time you'd seen something like this?
It is not the first time that I have seen a case of this type of organism.
Okay. And since then, you've seen it before again as well?
No, so this is I just saw this down there, and I
When you
have
were
seen
there
cases.
recently, right?
Yeah, and I've seen cases,
All right. You all set, Dickson?
Yep.
TWiP 97. You can find it at iTunes and also at microbeworld.org/TWiP. If you have questions or comments, send them to twip@twiv.tv, and please send your guesses to twip@twiv.tv. And we're going to get back in 2 weeks or so. Daniel Griffin is here at Columbia University Medical Center. Thank you, Daniel.
Oh, it's been a pleasure as always.
Daniel's about to hit the wards.
You are?
Yeah, I saw patients this morning, but next week's this time I will be over there attending on the inpatient wards.
How about that?
You'll be
The
doing
world can rest a little easier
infectious diseases?
It'll be the HIV/infectious disease service.
Okay. You will have plenty of work, I'm sure.
Yes.
Dickson Despommier is at Racaniello.org and verticalfarm.com.
Yep.
Thank you, Dickson.
You're welcome. It was
I always
a lot of fun.
have many sites to choose from. It's wonderful. I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiP is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
Is
parasitic.
parasitic.